Thanks, everybody, for joining us. I'm excited to introduce you to Steve Shallcross, who is the CEO and CFO of Theriva Biologics. Steve has been leading the company since 2018 and brings with him a strong background in operational and financial strategy. At the heart of the company's pipeline is VCN-01, a systemically administered stroma-degrading oncolytic virus designed to break down the tumor microenvironment and boost immune response. The company recently completed enrollment in their phase 2b VIRAGE trial for metastatic pancreatic ductal adenocarcinoma, a notoriously hard to treat cancer. We're looking forward to top-line data expected in the second quarter of this year, which is going to be a major milestone for the company. VCN is also being developed for retinoblastoma, a rare pediatric cancer, and has been granted rare pediatric disease and orphan drug designations in both the US and in Europe. With a strong foundation of science, multiple upcoming catalysts, and the next generation virus platform, including their proprietary albumin shield technology, Theriva is positioning itself to be a significant player in the oncology innovation. With that, please welcome Steve Shallcross.
Thank you all for coming this morning to hear the Theriva story. I'm Steve Shallcross, I'm the CEO. Theriva is an oncology company that is focused on developing, we think, the next generation of oncolytic viruses to be used in combination with other types of standard of care cancer treatments. And today we'll talk about the work that we've successfully completed and where we're headed. I will be making forward-looking statements, so I'll refer you to our securities filings for all the appropriate disclosures since we are a public company. So what are we doing that's different from other folks that are working in this space? So we think we're developing something quite unique. It's an oncolytic virus platform optimized for systemic delivery. And that's important because a lot of the treatments that are being developed today that utilize OVs are typically delivered intratumorally. And the benefit of using systemic delivery is that not only does the treatment target the primary tumor, but it goes after the metastases. And as many of you probably know, that's ultimately what kills these patients. VCN-01 is our platform technology. And we announced last fall that we just completed our enrollment in a phase 2b study in first-line metastatic pancreatic adenocarcinoma in combination with standard of care chemotherapy. We're expecting our top-line data in the second quarter. So it's just around the corner, and it's going to be a very, very exciting time for the company once that data gets released. It will help us chart our path forward. We've also conducted a number of phase 1 studies with leading institutions around the world and a number of indications which include retinoblastoma, head and neck cancer, colorectal cancer. And we've also put our treatment in combination with immune therapies like the meso-CAR T therapy from UPenn in ovarian and in pancreatic cancer patients. There's also a platform of new technologies in development, and we'll touch on that a little bit today. We're publicly traded. We're on the New York Stock Exchange. Ticker is TOVX. At the end of the year, we had a little over $12 million of cash in the bank with a runway that gets us into the third quarter of this year. We're a small company, 22 employees. We got 10 in Rockville, Maryland, and a dozen in Barcelona, Spain, where this technology that I'm going to talk about today was developed. We also have a BL2 facility in Spain, so we do a lot of our development and manufacturing work over there. This is our pipeline slide. Again, most of the financial and human resources that the company has are committed to the development of VCN-01 and the pancreatic cancer indication, and we'll talk about that at length. We just completed a retinoblastoma trial in pediatric patients. We'll touch on that. About a year ago or so, we had some data released in a phase 1 study in head and neck cancer combining our treatment with checkpoint inhibitor therapies, durvalumab, which is AstraZeneca's drug. And then we have an ongoing brain tumor study at Leeds using our virus. And this is primarily a PK study to see if the virus actually crosses the blood-brain barrier and can get to the tumors. I mentioned our development platform, and we also have some legacy products in 4. which is a product in development as a preventative in acute graft-versus-host disease in bone marrow transplant patients. That's being conducted at WashU. And then we have another asset, SIN-20, which is an oral form of intestinal alkaline phosphatase. This is for various inflammatory GI disorders, and we're in active negotiations to license that out. So what are we doing that's unique? Well, first we need to talk about the technology. So the VCN-01 platform is built around an adeno5 as a common cold virus. And what's special about this is that first and foremost, we're able to deliver it systemically, IV. And as I mentioned at the onset, that's good because we go after the primary tumor as well as the metastases. Delivering it systemically also allows us to deliver it in very, very high doses, which addresses the neutralizing antibody issue that often comes up with using viruses systemically. The second point here is that it's very, very selective. In other words, it's been designed to only replicate in tumor cells. It leaves all healthy cells alone. At the same time, it's been designed to target the liver. A lot of these products tend to be very toxic to the liver. We've addressed that problem and made it as safe as we possibly can. But more importantly, it's stroma degrading. And for those of you that know anything about these difficult to treat solid types of cancers, the problem why a lot of the treatments today aren't very effective is because the tumor cells are integrated into what we call this stroma or matrix. It's this fibrous basket of cells that prevent chemotherapy, checkpoint inhibitor therapy, immunotherapies from actually reaching the tumor cells and actively killing them. We've addressed that problem because as the virus replicates in these patients, PH20 or hyaluronidase is released, and as it's released, it starts to degrade this very fibrous, stroma, if you will, and I've got a picture to show you shortly to explain that in more detail. So how does this work? So here's a little visual. So here's a tumor surrounded by this fibrous material. So we introduce the VCN-01 treatment IV systemically. It starts to circulate. As it circulates, it goes, again, selectively only after the tumor cells. And it's able to replicate very, very rapidly and in very, very high quantities. We're talking like 1 million replications per tumor cell. So very, very effective at reproducing. As that happens, PH20 is released, and that's what you saw there by those virus cells that expressed PH20. That helps to break down this stroma or the matrix, and then gives us the opportunity to introduce the other typical standard of care type treatments. In this case, standard of care chemotherapy, which is the combination of nab-paclitaxel and gemcitabine. And then naturally, the body sees this virus and it starts to create its own immune response. So we can sort of get this 1-2 punch. We introduce the virus, we start breaking down the stroma, we start to see an immune response, we introduce the chemo. So now we got what we believe is a more effective treatment. The product itself has been in humans, it's over 142 patients in a number of different indications. Pancreatic cancer, both through systemic delivery and intratumorally. We've also had the product in retinoblastoma patients, colorectal patients, head and neck cancer patients. In addition, as I mentioned at the onset, in combination with CAR T therapy in pancreatic and ovarian cancers. So, as I'm sure all of you realize that pancreatic cancer is probably the most deadly cancer that we know about today. It's an orphan indication, and once diagnosed, the median survival is somewhere in the 8 to 11 month range. And that's primarily because that the tumor cells sort of integrate themselves around the vasculature. You can't surgically remove them, and then the only other options are various types of treatments that are available today. In the U.S. in 2024, there were just under 70,000 new cases diagnosed and over 50,000 deaths, and that continues to grow, and we're continuing to see that pancreatic cancer is starting to affect younger and younger people, which is very, very concerning. This graph on the On the right-hand side there is a resection of a part of a pancreas with the cancer cells integrated. You can see how it kind of looks rubbery. That's what we refer to as the stroma or the matrix. And this is why these cancers are so difficult to treat. This is primarily made up of hyaluronic acid. And as I said, our treatment was designed to release PH20 or hyaluronidase. It breaks down hyaluronic acid to make the treatments more effective and to produce an immune response. The market is very, very large, continues to grow. The latest projections we had, it's about $7 billion in 2030. We think it's going to well exceed that. So how did we get to where we're at today? A phase 1 study was conducted, and it's a relatively small study, about 42 patients, and it was done in 3 arms. The first arm was a monotherapy, and this was conducted primarily in colorectal cancer patients, and the purpose of this study was to evaluate safety and try to find out what the most tolerable dosing level would be for this product. Then we looked at 2 additional arms, arms 2 and 3, and these were specifically in pancreatic cancer patients. We wanted to see if it was more effective to deliver the treatments together or to separate them. So in arm 3, we delivered VCN-01 in combination with standard of care therapy all at once and looked at the results. And in arm 3, we delivered VCN-01 first, we waited a week, and then we started the patient on their routine chemotherapy regimen. So here's the data that's pretty remarkable. So first look at the right-hand side of the graph. This is the safety side effect profile. This is an adenovirus, which I described very early in the presentation. The most common side effects associated with this product were flu-like symptoms, pyrexia. These resolve in 5 to 7 days, and it's what you would expect by administering a virus to a patient. We saw some increases in transaminase levels, liver enzymes, but those fully resolved in 5 to 7 days. Remember when I said that when this was designed, it was designed specifically to target the liver, and here's clinical proof that in fact that has happened. But now let's look at the efficacy data, and I know these are small n's, but nonetheless, the green bar on the left-hand side, this is the data from a standard of care therapy, gemcitabine and nab-paclitaxel, that was conducted in a phase 3 trial in 431 patients. So we didn't do this. This was published data. When the data came out, about 23% saw a response. So what do we mean by response? That's a reduction in the primary lesions of 30% or more. So you say, oh, that's pretty good, but it's not as effective as one might think. More importantly, the median survival was about 8.5 months. Now look at the product in combination with the VCN-01 treatment. We had a low dose of 3.3 times 10 to the 12th and a high dose of 1 times 10 to the 13th. Total, we saw a 50% response rate and an overall 13.5-month survival. But there's clearly a dose response here, and you can see at the high dose here, the second column, that we saw about an 83% response rate. That's 5 of the 6 patients in that cohort, and about a 21-month overall survival. This was very impressive and gave us the confidence that this is a program that was worth it to advance further into the clinic. When we talk to our KOLs, there's a matter of measurement called the hazard ratio. Some of you may be familiar with it. The hazard ratio is important because it tries to balance out the various variables in the patient population that you're treating. It's the relationship between the overall survival in the treatment group versus the standard of care group. One is the same, lower numbers are better. Our KOLs say if you can get a 0.7 or lower, you have a viable product that would translate into improved survivability and better quality of life for the patients. This is some clinical data that was generated from the phase 1 study. On the left side, we were able to measure PH20 or hyaluronidase in the patient's blood. You can see day 0, obviously, there's nothing in the blood because the virus wasn't introduced yet. Once we introduce the virus, you start to see viral replication, and as a result, we can measure PH20, and that went out for 28 days as far as we measured it. In our head and neck study, we actually measured this out to 70 days. So as long as you can track pH20 in the blood, you know the virus is still replicating because tumor cells are still present. On the right-hand side, we see the immune response that the body is starting to generate, or an inflammatory response through a number of markers, CD8, IDO, and PD-L1. But what's quite remarkable here is that this data was collected from hepatic metastases, so from liver samples. So we know that the treatment is getting to other parts of the body that have the cancer, not the primary tumor. So this all gave us the confidence to advance the program forward in a phase 2 study. The study that we conducted was a multi-center, open-label, randomized controlled program. It was in first-line treatment of patients with metastatic pancreatic ductal adenocarcinoma, PDAC. These were all newcomers. A lot of phase 2 studies, you only get the opportunity to treat after standard of care treatment fails. These were first-line, newly diagnosed patients. They were randomized 1-to-1, gemcitabine, nab-paclitaxel as standard of care, or up to 2 doses of VCN-01 plus the standard of care. Primary endpoints, safety, tolerability, and overall survival. Overall survival is the approval endpoint that the FDA uses to grant approval for these types of products. We're also looking at response rate, progression-free survival, landmark survival. We achieved our target enrollment. We actually went over 92 patients. I think we enrolled 98. It was well balanced in both arms, and as I said at the onset, the study was conducted in Spain and in the United States. So we completed the study in September. We announced that. We had our first Data Safety Committee meeting in the first quarter of last year. There were no safety concerns raised by the committee. No protocol amendments were requested. We recently had a second Data Safety Monitoring Committee meeting, and we expect to put a press release out on that shortly on the outcome, but there should be no surprises there. Again, top-line data expected here coming up in the second quarter. We have orphan drug designation both in the U.S. and in Spain. We also received fast-track designation from the FDA, and we've already had multiple discussions with regulatory authorities, AIMS in Spain, the FDA, and the EMA, and right now it looks like they all are in agreement that the study design for the phase 3 should be essentially the same as the phase 2, with the exception that it would be fully blinded. And we're also looking at other opportunities with FOLFIRINOX and NALIRIFOX as possible candidates in future studies. So that's the pancreatic program. So I'll touch on our other 2 programs briefly. We, about a year or so ago, finished the study in head and neck cancer utilizing VCN-01 in combination with checkpoint inhibitors. These were patients that were refractory to previous rounds of checkpoint inhibitor therapy. In other words, they failed. They were expected to live probably 4 to 7 months. Again, we set the study up trying to see if it was better to give both of these treatments together or to separate them. Give first the VCN-01 treatment and then introduce checkpoint inhibitor therapy again. The other opportunity we had here is to get biopsies to do further genomic testing for these patients. The data for this trial were quite impressive. I'll just draw your attention to the light blue lines right here. Again, in the sequential arm, we saw the best response. At the low dose, we saw a median survival of 15.5 months. In the high dose, a median survival of 17.3 months. Remember, these patients were supposed to be dead in 4 to 7 months. So this was quite amazing. And the reason that you have the NE in there still is that we still have patients living. In this case, 4 years in both the low dose and the high dose. So something happened here that's quite unique. Again, these were refractory patients. We think by the introduction of VCN-01, the tumors were resensitized so that when they went on for subsequent lines of treatment with checkpoint inhibitors and then after that chemotherapy, we started to see a response. This chart on the right-hand side is something that the researchers like to look at. There's a CPS score, which is made up of a number of biomarkers and immune response markers, and they plotted that against the biopsies at day 8. So day 8, and why that's relevant, was the day where they received the VCN-01, but not the subsequent checkpoint inhibitor therapy again. And you can see for the sequentially dosed patients here that we have a statistically significant correlation here. So again, something here is going on that's quite remarkable. In summary, we've drawn the conclusion that the safety profile is quite acceptable, particularly in use with durvalumab. We know that the product is reaching the tumors, and we've been able to demonstrate that through the analysis of the biopsies that we've collected and the blood work we've run to determine whether or not PH20 was being expressed. And then, again, we saw this immune response in a number of the biomarkers that we have analyzed. So this work is expected to be published very shortly in a pretty reputable scientific journal. And once that happens, I think we'll have an opportunity to potentially talk to partners on how we further advance this program in the clinic. Next, we have this program in retinoblastoma. Again, this is a very, very brutal disease for these young pediatric patients, and right now there is no approved treatment for retinoblastoma. It's sort of the Wild West out there. The clinicians that treat this disease are using various forms of chemotherapy. They're either delivering it intravitreally, so directly into the eye, or through the retinal artery. So again, no standard course of action, and they're trying all types of things to try to save these patients' eyes. So it is an orphan indication. It affects 2 to 3% of the kids, both in the US and Europe. The numbers are very similar in Central South America and Asia. In the US and EU specifically, there's about 200 to 300 cases each year. And if you look at this picture here, you can kind of see how it looks in these patients. You see a normal eye here on the right-hand side of this child, along with the radiograph. You can see it's clear and normal, but on the right side here, or on the patient's left side, you can see this white pupil. That's being identified by this tumor right here that you see. So it's a brutal, brutal disease, and it's important that these patients get treated very, very quickly, because when chemo fails, the only resort is the nucleation of the eye, because they want to prevent the cancer from spreading to the brain. If that happens, it's a death sentence for these kids. So often, more than not, these patients lose their eyes. So we conducted a study. This study went on for a number of years, 15 years, since 2015, I'm sorry. And the reason is we were only allowed to enroll patients that were refractory, so they failed everything else, and this was sort of like the last-ditch effort. 9 patients were enrolled, ages 1 to 12. And again, they tried various forms of chemotherapy administered in a number of different ways. And we evaluated a number of different dosing regimens to try to find out if we saw better performance with a lower dose over the higher dose. There's 2 data points here selected, patient number 2 and patient number 3. And you can see at day 0, you got tumor, and these little specks are called vitriol seeds. Those ultimately result in tumors. Same thing here. This patient, patient 3, had a tumor here that actually calcified and stayed dormant, but they started to grow additional tumors. After the introduction of VCN-01, you start to see improvement, and in day 28, essentially all the vitriol seeds went away in this patient, and then they saw some reduction in the tumor sizes. But in patient number 3, this patient totally regressed. So they're told you, this patient is going on over 3 years now and actually using that eye. So that was quite impressive, and you can obviously see why there's a lot of interest around this program. So in summary here, we've only commented on 6 patients because the trial just recently finished. We will have the full data set available for public review coming up at ASCO in June, so it'll include all 9 patients. So we just got notice that we've been accepted to present the data at ASCO. But for the first 6 patients, we saw a reduction in vitreal seeds in 3 of the 6 patients. A nucleation was avoided in 2 of the patients. And the eyes that had to be enucleated, we think it was primarily due to the damage that the chemo caused. So we didn't really have an opportunity to fully evaluate these patients because sort of this is their last opportunity. But again, we see this as an encouraging opportunity for us to advance this program and either use it alone or in combination with standard care chemo. We're working with world experts around the globe, including South America, Spain, and even here at the U.S. We've actually had collaborations with St. Jude's. So another very exciting program that hopefully in short order we'll be able to advance. Real quickly, here's some safety data. I'm not going to dwell on this in addition to some expression of viral protein data, but this chart basically says we saw some inflammation as a result of administering the virus in the eye. It was well tolerated. We saw a little bit of turbidity or some cloudiness, and we treated that with standard of care steroids, and it seemed to work. There was no damage done to any of the healthy cells in the eye. So again, another demonstration that we think the safety side effect profile is quite attractive for advancing this program. So I mentioned we completed the enrollment. We're continuing to analyze the data. This data will ultimately be published outside of the presentation that's coming up at ASCO. We think that designing and using this in combination with standard care therapy is probably the best option for these patients. How it's delivered is still something that we're debating with the clinicians, whether it goes intravitreal or through the retinal artery. We've also received orphan drug designation for this product in the U.S. and EU already, and we've had 1 meeting with the FDA, and once we have a protocol that we can reach agreement on with our KOLs, we'll go back to the regulators and have another discussion with them. We also recently received rare pediatric drug designation from the FDA on this asset as well. Finally, we'll talk about our development platform very quickly. We have a new backbone that we've added to our treatment. It's called the albumin technology shield. There's been a lot of concern in the space where OVs are used on whether or not neutralizing antibodies, in other words, your body's own immune response attacks a virus once it gets introduced into your body. We don't seem to have that problem because of the high doses we're delivering it in, but what we're interested in is the delivery of multiple doses. So how do you prevent the body from responding to that? Well, we've come up with a clever method to use the body's own albumin in their blood to protect the virus as it circulates and goes after the tumor cells. So we're conducting a number of animal studies, and after that we'll be able to advance this program forward. More importantly, we're looking at a number of additional payloads to introduce to our backbone technology, and we hope in the next quarter or so to make an announcement on the product that we hope to advance further into the clinic. We're looking at some interesting things with various forms of hyaluronidase that could be more potent, as well as the introduction of PD-L1 type technology or toxins or parasporins. So something to stay tuned and watch. Very exciting. The goal here is to ultimately come up with a monotherapy so we don't have to use this in combination with other types of treatments. So what do we have to look forward to? We had a busy end of 2024. We had a busy first quarter. Going into the second quarter, as I said at the very, very beginning today, we plan to announce top-line results for our phase 2 study. That's a pivotal event for the company. We hope to have a protocol designed for the retinoblastoma as well as the presentation that we plan to make at ASCO in June, and then select our VCN 12 candidate. This is the next generation of product. In the latter part of the year, we're producing a lot of manufacturing activity as we scale up the production of our product, so we'll have some announcements on that. Again, the retinoblastoma design. And we think the cell line that we've developed for making our product can be quite helpful for other companies out there. So we're looking at additional ways to commercialize that. That's it for today. We've got a lot on our plate. Thanks for your attention, and I'd be happy to take questions or afterward talk with anybody in the back. Thanks again.